Hello, and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today we'll be talking to Chelsea Duball about the environmental impacts of oyster farming. What is oyster farming? How can it affect the local ecosystem? How do those changes affect the soil? Answers to all these and more coming right up. I'm your host, Abby Morrison. Let's talk about science. Hi, everybody. Welcome to the show. Today we have Chelsea with us. Chelsea is a PhD student in soil science at the University of Wyoming. She previously attended the University of Rhode Island, where she earned her bachelor's in environmental science and management with a minor in soil science. During that time, she pursued several internships focused on coastal and wetland ecology in places such as Puerto Rico and Narragansett Bay in Rhode Island. Upon earning her bachelor's, she continued on at the University of Rhode Island to earn her master's in biological and environmental sciences. Her thesis research was aimed towards understanding the impacts of oyster aquaculture on subaqueous soils and in fauna in Rhode Island's coastal lagoons. Hi, Chelsea. Welcome to the show. How are you doing today?
I'm great. Thank you. How are you?
I am doing so well. So happy to have you on the show today. So today we are talking about oyster aquaculture, which is oyster farming. So can you just give us kind of a high-level overview? How does oyster farming work? How do you do it? What does it look like? And why is this something that people are seeing a bit of a boom in? What are some of the benefits of oyster farming?
I'm happy you asked. Starting this research, I actually asked myself all of these questions. Coming from a soil science background, I used to look at cornfields and soybean fields, and now I was looking at how to grow oysters. So to figure that out, I did a lot of research. I went and I talked to the farmers themselves. But essentially, it's a crop. Oysters grow in confined environments, either cages, bags, located in these coastal lagoons, roughly 8 to 10 centimeters above the soil surface, if it's not floating aquaculture gear. And they just sit there. They're naturally a sedentary species. And what those oysters are doing over time is they're filtering the water within the coastal lagoon environment. And as they do that, they're filter feeding off of the particulate matter and nutrients that naturally occur in those waters. And that's why oysters are praised as ecosystem engineers. Oysters themselves can filter up to 48 gallons of water per day, and that's for a single oyster. And in that process, they can clean that water. But on the flip side of that equation, as they're taking in that water and those nutrients, metabolizing it into their shells and tissues, they expel whatever their bodies aren't using in the form of biodeposits, otherwise known as oyster feces. And so that's where our research came into play to see how aquaculture practices, how those oyster biodeposits were affecting the underlying soils under the aquaculture gear. And this is something that really had not been studied before.
Sure. And when you're talking about this gear, I mean, how are they getting these to float? Is it just like these little buoys that float around in a fenced off area of the lagoon? Is it they're strung from like some big net? How does that work?
So it can be both ways. The gear can be suspended by buoys themselves or nets. There is no fenced in area because these are state waters. So within the lagoon itself, they don't want a fenced-in environment because it's kind of an eyesore to local communities and any recreation that's also occurring within the ponds. So that itself, it doesn't confine the exact lease area. Most of the gear isn't highly visible, especially if it's underwater and not floating. So definitely the buoys. If it's not floating gear, the cages are made out of heavy metal, so they just lie on top of the soil environment.
That sounds super cute. First of all, I did not realize that. A strange definition of cute. It's fine. So I, first of all, did not realize that there was like recreation happening. So it just makes me really happy to think of like kids splashing around or people boating or, whatever they're doing in that area with these little oysters just chilling under the surface. Also that they're like not fenced in, like they could make a run for it if the tide is right. It's just really, really fun for me. And also I'm sure it just showcases my lack of knowledge of how lagoons work. But I love it. I think it's great. So thank you so much for that overview. So getting back to your research though, you were investigating maybe some of those potential drawbacks. So can you tell us a little bit more about what was your hypothesis and what specifically were you checking for in your research?
Yeah, of course. So in this study, we performed 3 major experiments. The first was to collect oyster biodeposits from different sized oysters at varying stocking densities, meaning the amount of oysters per given rack or bag within a given area. And this was going to help us better understand the amount of biodeposits produced from aquaculture systems as a whole, and what the nutrient or nitrogen and carbon content of those biodeposits were. This is essentially considered the impact of the aquaculture to the underlying soil environment. From here, we wanted to take it a step further in order to look at how those biodeposition rates and the content of those biodeposits were affecting the soil environment over both the short term and the long term. In order to do this, we conducted 2 additional experiments. The second being that we focused on the short-term impacts of oyster aquaculture by investigating how biodeposits impacted soils over a week-long period. To dive into that a little bit deeper, we looked at the impacts of varying stocking densities. So how many oysters within that given area could affect those soils within a 1-week period. The third experiment focused on the impacts of aquaculture over the long term. And to do this, we utilized pre-existing aquaculture sites that were active, and the age of use ranged from 3, 6, 8, 12, 13, and 21 years of use. So a pretty good range there. The objective of that long-term study was to investigate how the aquaculture practices impacted physical and chemical soil parameters and the communities of critters, or those infauna that we previously mentioned, living within those soils. And this was across 0 to 20 centimeters in depth. So from these experiments, we expected to observe significant negative impacts at both those higher oyster stocking densities in the short term and at older age sites. And this was as a result of higher biodepositional loading rich in those nitrogen and carbon inputs. The reason we predicted this is because over time, those biodepositional loads are capable of over-enriching benthic environments, or those soils lying beneath the aquaculture gear.
Yeah, so one of the things that I found most interesting about this, so for one thing, I love animals. So like any kind of animal research related research is I'm into it. I want to know more. So something that I found really unique about yours though was these infauna. So in these kind of underwater subaqueous soils, you have these creatures. Can you give some examples of what kinds of creatures they are, how big they are, what they look like, anything like that?
Yeah. So these creatures had a wide range. A lot of them were worm type animals. They ranged in size from a fifth of my thumbnail to the whole size of my index finger. And essentially, some of these look like aquatic centipedes. They have tons of legs. They have huge pinchers at the end of their mouthpieces. And those are typically polychaete worms. There's other worms as well that don't have any legs, and those are oligochaetes. So some big words there, but essentially worms with legs or worms without legs.
That's awesome.
Yeah, and there was a whole variety. We found some smaller anemones, smaller shrimp or amphipods. Each soil that we sieved out, we found some new surprises.
That's awesome. That makes me just want to dive down like a Google search rabbit hole.
Oh, we did.
Yeah, I'll bet. Did you work with any like marine biologists maybe to identify them, or did you not care like what kind they were so much as just that they were there?
We actually did collaborate with quite a few marine biologists and oceanographers at the University of Rhode Island's Bay campus, as well as folks over in the biology department. It took about a full year going back and forth to hone in on what creatures we were looking at, as well as what sieve size to use, meaning smaller creatures will fall through a large sieve size. So we really wanted to maintain a good sample. So we took a while to figure out what that should look like. I should note I'm very fortunate that I had over 5 interns in this process, and one intern in particular was very well skilled in identifying marine macroinvertebrates. So I am very thankful for Lauren Salisbury.
Wow. Go Lauren. What a very specific niche to fill. That's great. So moving on then, so we had these 3 different experiments set up to kind of test the different ways in which these oysters and their feces can affect their surroundings. So the first one you mentioned, you were really just trying to start collecting the fecal material and see kind of how much there was. So the keen listener is probably thinking at this point, it's underwater and probably very small. So how did you even go about doing this? How did you know that you were collecting the right amount of stuff, that it wasn't just getting washed away? Tell me all about that.
Totally. We had our doubts going into that whole process. We framed our collection methods off of a few previous papers. So we were fortunate those existed. So really, we just went out to a farm one day with a couple of old tin cake pans and zip ties, and we stomped out onto the farm. The farmers were there that day, and they said, "Chelsea, what are you doing with those pans?" I said, "I'm going to collect some oyster feces." And they said, "Usually we want to get rid of those." So I dove into the water, threw myself under some of those racks, zip tied those pans, left them out there for a day, up to 3 days. And lo and behold, we came back after that day period, and we had some nice mucky particulate matter that had accumulated under these racks. And it was clear that it wasn't sand. It wasn't, aquatic plants. We had actually caught those biodeposits that we were even wondering if they existed in the first place.
Wow, that is crazy. Okay, I'm going to ask perhaps what is I'm not going to say stupid question. It's a valid but clearly uninformed question. Can you explain more about just the physical setup of these lagoons or other water environments that you were investigating? I mean, are there tides or currents that could really affect what you're doing? How deep is this water? Obviously, if you were diving, I mean, it's not super duper shallow, I guess. Can you tell me more about that?
Of course. So these coastal pond areas are highly favorable for recreation because they are fairly shallow environments. They're considered brackish waters. Each coastal lagoon is connected to a breachway, which connects out to the open ocean. So essentially, the barrier between the coastal pond and the ocean is what we call a barrier beach. And just behind those barrier beaches is where we set our sites, located on what we call a washover fan. And the waters located on top of that washover fan area were about 1.5 to 1 meter deep at high tide. And that leads into the next point here, is that these areas do experience quite a significant tidal range. However, they're not fully drained at any point.
Yeah, Yeah, that's super helpful to understand more of what you were looking at here. So you started collecting your pans, and we will talk more about the results of kind of that initial experiment in a bit here. But then you were also trying to see kind of that short-term impact. So can you tell me how you went about doing that experiment?
Yes. So kind of riffing off that first experiment, we established what those biodeposition rates looked like per oyster and within a given area occupied by the aquaculture gear. And using those numbers, we were able to come up with biodepositional loads that represented essentially stocking densities of high amounts of oysters, an average amount of oysters, and no oysters within a given area. And we took those biodeposits, we froze them, and then we applied them to areas within the coastal lagoons that had never been used for aquaculture before. And what we were seeking to test here was if we were to, say, establish a whole new aquaculture farm in an area that had never been used for aquaculture previously, how would that impact the soils in that area? And because we were only looking over a short-term period of up to 1 week, we just sampled the upper 2 centimeters of those soils to figure out if there was enrichment of nitrogen or carbon from that high to average amount of oysters in that given area that we had applied artificially.
Okay, so then you were able to do this short-term experiment, kind of checking to see the impacts there. And then you wanted to extend that. Can you tell me how you conducted the long-term experiment?
Of course. That's actually where we started with this whole idea, is to look at those pre-existing aquaculture areas and how they had affected those soils and fauna within those soils over time. So in order to do this, we had to find some oyster aquaculturists that were willing to allow us onto their leased areas to manipulate some of their racks and take some big soil cores over the course of 2 years. To find suitable areas that were relatively the same across 3 coastal ponds, we utilized some Google Earth maps. There's a really cool time hop feature within it, where we went back to aerial imagery dating as far back to 1992, all the way up to present day, which was 2014 when we first started establishing these sites. So that's how we went in and we found each of our sites. And as I had mentioned, we found sites that range 0 to 21 years of aquaculture age. And from there, we went and we collected our soil cores in order to look at those different soil parameters. And then we took additional cores where we sieved them out to get our infaunal samples.
Hi everyone, I hope you're enjoying the show. Interested in learning more? Chelsea's article, Impacts of Oyster Aquaculture and Subaqueous Soils and Infauna, published in the Journal of Environmental Quality, will be freely available for the next 2 weeks. You can find a link to it in our show notes. If you are a certified crop advisor or certified professional soil scientist, you can also take a quiz for continuing education units for this episode, which can be found in our show notes or on certifiedcropadvisor.org. Let's get back to the show. All right, so you did all of this work for these 3 experiments. What did you find?
That is a great question. So that brings us back to the 3 different experiments here. The first being, are those oysters actually producing biodeposits? Can we catch those biodeposits? And then from both of those, can we come up with a number of how much they're producing? And what we found from that was larger oysters produce more than a smaller oyster, which is fairly obvious in most cases. But they were producing a significant amount of biodeposits, and we could actually measure that. So that was a first very important finding for us. That then allowed us to conduct our second experiment where we collected a whole lot of those biodeposits, again, froze them, applied them to those soils, but across our different treatments, being a high stocking density, an average stocking density, and then a control, meaning no oysters were in that area, we didn't see any differences in nitrogen or carbon accumulating within that upper 2 centimeters of soil from 0 to 7 days, which was really fascinating to us because we thought we would have seen something, particularly in that high stocking density, because we had put a very large amount, equivalent to 2,000 oysters within a square meter.
Wow.
So that it was fairly shocking. So that got our gears turning a little bit. Were these floating away? No, we had accounted for that in our methods. It just went to show that these soils are capable of processing those high nutrient loads. And this could be due to infaunal processing, so the critters feeding off of some of that material, or translocating it, meaning burying it deeper down into those soils, so past that 2 centimeters. But one thing that's really fascinating was this had occurred over just that 1 week period, which was great to see. Leading us into that third experiment, we wanted to know what was happening over longer periods of time. And so those sites, again, range 0, meaning our control site that hadn't been used for aquaculture, up to 21 years of age. And what we had found across these sites was no significant impact of age on any of those chemical or physical soil parameters that we had looked at. However, sites that were 5 years or older did have very specific differences in the infaunal communities. And so what we were seeing there was higher amounts of what we call deposit feeders or opportunistic species. And so from that, we were seeing higher populations of both of those, again, in aquaculture sites that were 5 years or older, compared to those control sites that had never been used for aquaculture before. One thing we speculated here was those opportunistic species were coming in and feeding off of the higher amounts of biodeposits that were being produced under these aquaculture racks. And that could have been a fairly obvious prediction. However, we weren't seeing higher nitrogen or carbon contents within those soils. So the soils weren't over-enriched in these materials. And that could be because those infauna, or those higher amounts of opportunistic species that were there, were processing that material, which was pretty cool to see.
Wow, that's super neat. I like that.
Yeah.
So now that we know kind of that you found that oyster aquaculture doesn't seem to have nearly as negative as an impact as you were maybe expecting, I kind of want to change gears here a little bit and talk more about some future research here. And there's a little bit of maybe two paths to take here. We can travel both if you like. So the first would obviously be just what is the next step for this kind of research about the impacts of oyster aquaculture, anything like that. But also, so this is branching off kind of from actually a 99% Invisible episode about oyster aquaculture. They called it Oyster Texture because they were talking about it in an architectural sense. What are oyster aquaculture's kind of seen this weird, like, new use resurgence kind of thing. So the other thing I wanted to talk about here is just what are some of the benefits you see for maybe increasing or using oyster aquaculture whether that's in climate change or protecting shores or anything like that. So those are kind of the two questions I have for you.
Yeah, those are both excellent questions. One thing to note is this study focused on oyster aquaculture, so not wild populations of oysters that create those notorious reefs along shorelines or anything like that. But there's more studies popping up, and our study contributes to this idea that aquaculture serves its purpose in acting as infrastructure for our ecosystems. So you don't need a wild reef in order to create habitat for other fish or critters that are swimming along or crawling along the soil surface in these environments. The gear itself can act as that infrastructure, similar to an oyster reef. So future studies should really focus on the differences potentially between those two types of oyster infrastructure. I think that would be extremely valuable, particularly when we think about climate change and increased storms and impacts of those coastal storms in these environments. What role do these aquaculture areas have in protecting some of that ecosystem? I think that's really important. Two is back to this idea of water quality in these environments. We know that these oysters are ecosystem engineers, and several articles highlight their capability to improve water quality. But going forth, we need to look at that issue from both, as we mentioned in this study, the water column and what happens on the other end with the oyster biodeposits. But what about the oysters themselves? Looking at the shell and the tissue of that oyster and seeing how much nitrogen or carbon and in other matter are they actually consolidating into their bodies? Because when you think about it, that oyster is growing in these waters, but they're growing for the purpose of being eaten. And therefore, they're harvested out of that system and taking some of those materials out of the system as well. Is that a good thing? A bad thing? I think it's leaning more towards the positive direction.
I hadn't even thought about that. Yeah, I mean, I know it's farming, so I don't know what else they would be growing for in that scenario. But yeah,
Right.
That's another fascinating aspect that I hadn't even thought about yet. Those are all really, really fascinating areas of future research. I also wanted to ask, what are you studying these days? Because as I believe we had discussed, you have kind of moved away from this a little bit. Is that correct?
I have. I've probably moved as far away from this as I possibly could. I'm now pursuing my PhD at the University of Wyoming, which is much drier. Everyone likes to make the joke that I will not find any oysters out here. But I do find similar soil properties in salt-affected semi-arid and arid wetland systems. So that's where my research is focused right now.
Wow, that sounds super interesting. Partly because you just said semi-arid wetland, which just seems weird to me.
Like an oxymoron.
Yeah. So that's in itself is fascinating.
Yeah, they are extremely valuable systems that most people don't think of as often. But when we do start to think about it, wetlands are extremely important in these semi-arid and arid systems because that's where the water is. And that's very important for flood abatement, habitat for migratory species like sandhill cranes, increased biodiversity. So they're almost serving as hotspots or valuable hotspots across what most people presume is a vast, dry, desolate area.
Right. Yeah. That is wild. Such fascinating ecosystems, both of these, really. That makes me so happy. So I've got 3 questions left for you. I'm going to restrain myself from just diving down into a whole podcast worth of wetland ecosystems. But maybe we can have you back on the show another time. For this episode, what if you had like 1 or maybe 2 key resources for people who wanted to learn more about this? Where would you send them?
Ooh, great question. A few key resources. Well, to get involved, listeners can totally support their local farmers, and that includes your aquaculturists. Acknowledge these people who are catching those fish and oysters and raising them on their own farms as your local farmer. You can also get involved with oyster restoration projects through the Nature Conservancy, such as the Billion Oyster Project. I think that's occurring in New York City. Or you can simply read up on the benefits of sustainable aquaculture on the environment, as well as, the whole process of going from that coastal pond or the open ocean to your plate. Maybe this will help you consider your diet and your impact on both the economy as well as the natural systems around you.
Yeah, totally. I also wanted to pitch in. This is rare, but I also have resources to offer. So one would be that 99% Invisible podcast episode I mentioned. It's called Oyster Texture. And I will include a link to that in the show notes, as well as everything you just listed. And then also recently, the podcast One to Grow On recently did like a guest show, maybe would be how to describe that, with a podcast called Sorceress, spelt like source, not like the typical like magic user. And they did one that was all about oysters as well. I haven't listened to that one yet, but I'm sure it's great. And then, yeah, so we kind of covered then how to get involved also in that question. Do you have any other tips for people who want to get involved with this?
I've actually started to, on the side, help a lot of friends and consulting for them as they start their oyster farms. I participated in an aquaculture class that was at Roger Williams University in Rhode Island, and it was an excellent effort that brought together community members, students, folks working with the local as well as federal government who wanted to get involved with oyster aquaculture. And it took you through the biology, how to start a farm, everything involved. But one thing that they didn't mention was the soil, which was fascinating to me. So my advice to anybody getting involved, particularly in shallow coastal environments, is to take an ecosystem approach and realize that these oysters, while they do need to feed off of those natural waters and those clean natural waters, they do need those soils to lie upon if you don't have floating gear. So there are resources out there. There's the Web Soil Survey. You can go through, at least in the state of Rhode Island and several states along the East Coast, they've mapped subaqueous soils in a lot of environments, and they're starting to expand those efforts to the West Coast as well as further down south in Gulf area. So take a peek at those soil maps. They're always fun to explore, but even more so in some of these coastal and marine environments.
Yeah. Wow. Look at you being a consultant.
Yeah.
That's so awesome.
I actually have a friend who we took that class together. He made his plan the next day, and I noticed he didn't think about what was under his oysters. I said, let me show you something. And we sat and we had a coffee, and 3 hours later, we had mapped out his whole farm. And here we are, 2 years later, he has such a successful oyster farm business. He's been at festivals all across the East Coast, and he's just really seen success. And I think it's because he looked from the ground up to plan out his whole business.
Wow. That's amazing.
Yeah.
We will have your contact information in the show notes. So anyone who wants a master consultant, please reach out. I'm just volunteering you to be reached out to. So last question. We've already had so many fun facts on this show. But aside from being master consultant, what is one fun fact that people would not know about you if all they had was your research?
Oh, one fun fact. Well, when I'm not digging in a soil pit or bobbing for oysters, you can likely find me experimenting new baking recipes in my kitchen. I probably bake every day.
Wow.
That's my creative outlet. Or, you can find me designing new artwork for my science communication endeavors.
Wow. What kind of art and also baked goods do you make?
I'm a huge scone fanatic, but I've been getting into breads lately, which is a lot of trials and tribulations. Something that comes a little bit more naturally to me, though, is drawing. I've really taken a knack into drawing over the years, and my focus has been on soils lately. Draw what you love, and I tend to do that. So I focus on geometric patterns to convey both aesthetically pleasing soils, but also accurate soils. However, the public communities can actually understand what's going on there. That's my main goal.
Yeah, a worthy goal.
Yeah.
That's amazing. That's so cool. Gosh, wow. Well, thank you so much for being on the show. This has all been so interesting. Thank you so much for taking the time.
Thank you, Abby. This has been great.
Thank you for listening to Field Lab Earth. You'll find a link to today's paper and other resources for this episode in our show notes or on our website. If you have any questions, comments, or recommendations for show topics, please contact us at podcast@sciencesocieties.org or on Twitter @FieldLabEarth. If you'd like to hear more content like this, please subscribe, and don't forget to rate and review us on Apple Podcasts or anywhere else you find your podcast if you like our show. This podcast is a joint production of the American Society of Agronomy, Crop Science Society of America, and Soil Science Society of America. Special thanks to Lobo Loco for the use of their song Spook Castle on the intro and outro of our show. Opinions and conclusions expressed by authors are their own and are not considered as those of the American Society of Agronomy, Crop Science Society of America, Soil Science Society of America, its staff, its members, or its advertisers.